So welcome to Bev and Alex. We've got some really interesting questions today, and we want to start with one about culturing. So this is really talking about cost and time with micro propagation and culturing. And do we know any ways of using different agars or cheaper ways to do it, or quicker ways to do it? Bev, do you want to?
Yeah, I mean, obviously, if you're growing things, you have to give time for cells to divide. So from that point of view, it's hard to make it quicker. In terms of alternatives to agar, I had a little look around, and there seemed to be some possibilities of using things like alginate or guar gum, or any other sort of gummy, starchy thing that you can sterilize. But obviously alginate is the best. You know, I even found somewhere that had tried using arrowroot and aspic and cornstarch to make a gloopy solution. But of course it wouldn't gel, it would have to be very concentrated. So I'm not sure whether those experiments were unsuccessful, which is why we just use agar at the moment. And as I say, in terms of getting things to grow faster, I don't think you can, but I think what the SAPS protocol does, and Alex, perhaps you want to come in on that, means that although you have to leave it, it's going to survive because it doesn't go moldy.
Yeah, I would agree with you on both those points, Bev. I think, I mean, the SAPS protocol particularly, you know, that technique was developed based on how micropropagation is done in the field. So obviously, in that situation, agar has been found to be the most successful, almost, or indeed most practical thing to be using. And yeah, the idea with the sterilization is that you would be able to have it, yeah, you would be able to leave the containers for a while and without them going moldy, because you've got that sterilized material in there. And in terms of other, you know, the not so much micropropagation, but the other cloning techniques, just the taking a cutting, they do take time to grow, but you should start seeing, you know, some root growth within a couple of weeks. It depends on the plant, of course, but yeah, not much really, unfortunately. Certainly at this at this level of being able to speed things up, particularly. So I suppose it's about how you plan that in.
In terms in generally using plant material, a lot of the SAPS practicals use supermarket plant material. So, you know, as long as you can afford to go and buy a bunch of flowers or a couple of basil plants or some, you know, a bunch of something to section, then, and you can make that do several classes, then you don't have to wait to grow seeds. You can actually obtain them all through the year, herbs particularly, to suit the needs of your classes.
Yeah, yeah, absolutely, Bev. That's a really good point. That you don't need to grow everything yourself to do plant practicals, and all of our practicals are developed to sort of be as efficient as possible, both with resources and with teacher, technician, and student time availability.
Thanks, some really good tips there. We'll move on now to some subject knowledge questions. Jeanette asks, is there a point in time when plant material in test tubes will stop respiring?
Yes, yes, when it dies. You know, if you're going to put a piece of pondweed into bicarbonate indicator, it will survive for a long time, as will algal balls. They can keep going for months, but eventually they will stop. But in terms of running these practicals for several classes, I should imagine check me out on this, Alex, but I should imagine your pondweed would stay alive for several days in the bicarbonate indicator. The algal balls will live for months in the bicarbonate indicator, but I think the pondweed in a closed system would survive for a couple of days. Over to you.
Yeah, I think you're probably right. I wouldn't be able to tell you. I haven't sort of tested how long it would survive in the bicarbonate indicator. But the pondweed, once that's cut off, I mean the cuttings will keep, but they won't keep as well as, you know, if you've got a whole plant, particularly if you've got a rooted plant. Obviously that's sort of rooted into the substrate. They'll survive much longer. But in the experimental conditions, yeah, they will start to degrade after a while. But the algae do, as Bev's right, the algae do survive much longer, and of course they are free living within the water under normal circumstances, so they are much more suited to being freely floating, whereas pondweed, for example, is, you know, a living pondweed plant is rooted into the substrate of the soil, so at the bottom of the water course that it's in. So without that, in the same way that any other cutting will eventually die, because it's not, or any other, sort of like a cut flower, for example, won't just keep surviving in water. That would be similar for the pondweed. And again, for the other, we had the leaf trapped down the side of the test tube, trapped by the bung, and that will, again, it's not getting nutrients from the nutrients it needs from elsewhere in the plant, so it will eventually, in that situation, break down and stop respiring. Yeah.
Thank you for that explanation. We've got a question as well from Donna. Really interesting question. Is it true that a Venus flytrap will die if it opens and closes too often without an insect inside?
Yes, that is the case, but it's not actually to do. I think there's a misconception around whether that's to do with, because it's not getting the nutrients. So Venus flytraps supplement their nitrogen intake with trapping insects, but actually a Venus flytrap doesn't rely solely on insects for its nitrogen intake. So it's actually to do with it's to do with more with energy usage in terms of shutting the trap. So it takes quite a lot of energy for the plant to shut the trap, and so for it constantly to keep shutting its traps, you know, if it's being bothered lots and lots of times, then that can be a problem. But for example, if you're just demonstrating the trap shutting, as we do in the SAPS video, that's not an issue, you know, to demonstrate that. So the can a Venus flytrap count investigation, that's not going to harm your flytrap. If every single, you know, if the traps are constantly being triggered, then it uses a lot of resources up for the plant, and then it's also not getting anything out of it. But it's not because they can't survive, or my understanding is it's not because they can't survive without the nitrogen supplement from consuming the insects.
From a classroom point of view, that does mean that you must make sure that while you want to encourage your pupils to be fascinated by your Venus flytraps, you don't want them flytrap bothering. You don't want them constantly prodding them. So you'd have to have some degree of understanding, I think, about that, explained to the class.
Thank you. We've got a question now about water molecules, or the description of water molecules when you're explaining osmosis, from Mrs. Reichnowski. Is it a good phrase to use, the concentration of water molecules, or should we be saying fewer particles of water or more particles?
Okay. Shall I start with this one? When we were writing the text, we decided that we would pitch it at GCSE level, and then obviously we consulted the exam board specifications and the exam board approved textbooks to sort of help us decide what kind of vocabulary to use. And concentration of water molecules is referred to a lot in those sources of information. And yes, I understand it is almost counterintuitive, because you're really talking about concentration of solute when you're talking about concentration of anything. However, that particular piece of information, of course, is intended for the course participants to use as a refresher. And we were trying to pitch it at participants who may be new to teaching or new to teaching biology, and therefore their background in osmosis isn't as extensive as somebody who's been teaching for a very long time. So that concept was picked on because it was felt they will come across it in their teaching when trying to develop their own resources. And of course, as with everything on the course, how you use the material, you pick it up and modify it to suit your teaching. You know, I know in the past people have said, why didn't you use water potential from the word go? But water potential is a concept that the students are introduced to at A level, not at GCSE. So that's why we picked that particular concept. But we would expect everybody to mould the information and express it in the way that's meaningful to them and their students.
Thanks, Bev. We're going to move on now to fieldwork. A few questions about fieldwork, and this started off quite a bit of conversation actually, so I was really enjoying reading some of the ideas that were shared. But Rachel would like to know, do you have any suggestions for the way you could use fieldwork to learn about other plant topics? So perhaps plant defense responses or stem cells or osmosis? Who wants to take this question? Do you want to come to you, Alex?
Yeah, I mean, I think Bev and I probably both got some ideas on this. I think it's I'm really pleased that that's come up actually in the discussion, because I'm certainly of the opinion that, you know, drawing a student's attention to these phenomena in the natural world that they would just see around them, even if we're not talking big field trips, but, you know, the plants that they might see in their school environment or their home environment, or on their journey, you know, between the two places. It's a really great way of adding some context for those students about how those things are relevant in the things that they see in everyday life, not just the examples that they might see in the classroom or that they might feel that they need to learn for their exams. So it's really good to see that come up. I think trying to think of some specifics now, but certainly one of the things that we often do with SAPS in terms of plant disease, we often do when we're training people to do, we have ambassadors who go out and talk about SAPS resources with other schools, and also we work with teacher trainers, and one of the things we do is sort of go for a walk looking for plant examples of plant disease, which is something that you can learn to spot the signs of plant disease in the world around you. So certainly students could very simply just go and gather examples of what they think might be examples of plant disease, and often the markings that you might see on a plant would be evidence of a plant defense. Or they could go out and gather, if you're looking at a defense such as having a cuticle, they could go out and, you know, a thick waxy cuticle, they can go out and find examples of those in their own environment. And again, looking for things, something like stem cells or meristems, you could look at, perhaps look at plants that have been pruned and things that have been cut back. which might be if you're in a more sort of maintained landscape rather than you might see that. Or thinking about if you are in a more rural setting or somewhere perhaps where there are animals that are coming and eating plants, looking at seeing what the effect of the damage has been. How has the plant responded to that, and seeing the new growth come from those meristems, and how that's a part of the way they're adapted to live in that environment. Have you got any other examples, Viv? I could probably go on forever thinking of things you could look at.
Yeah, well, I thought of a couple. So I thought of the SAPS practical looking at prickles on holly leaves in predation, and the students investigate, they count numbers of prickles at different heights up the holly bush to investigate whether or not, to investigate the hypothesis that grazing stimulates prickle production as an anti-grazing strategy in the plant. Also, there's the infrared thermometer investigations, which you can do in the classroom with basil leaves, but you can take infrared thermometers out and investigate various aspects of leaf surface temperature in response to the environment. And I was in my local builder's merchant last week, and they had tiny little infrared thermometers for £3 each. Tiny ones, so they're very readily available. And then the other one, as you say, just walking around, if you're going to do an ecological investigation, and asking interesting questions like, why are those buttercups only found in that damp soil? Why aren't they found in the wet soil? And then thinking back to how those particular types of buttercups might be adapted to that, maybe looking at sectioning their stems, looking at their root growth. So walking around and getting your students to ask interesting questions, and then going away and trying to think of how their physiology may be adapted and ways you can investigate that is a really exciting thing to do.
Thank you both of you, some really great ideas there. Well, that's all the questions that we've got. So I don't know if there's anything else you want to add, anything that's popped up during the course.
It's just been great. I've really enjoyed all the comments that the course participants have made. And I love it when I see the course participants interacting and answering each other's comments. There's lots of sort of information sharing going on, and I've really enjoyed it. So I'd just like to say thank you to everybody who's taken part in the course this time. It's been really interesting.
Yeah, absolutely. Yeah, very similar for me. Sorry, I spoke over there, Karen. Yeah, just it's always you know, we can see the numbers of people doing the course, we can get those stats, but the comments, it's really nice to see people reacting to things. We always, well, I certainly always learn things from the participants on these courses, both about how they're doing things in schools, but also, you know, some plant science things that I wasn't aware of. So it's always really, really nice. So again, yeah, thank you for all those, and thank you for the questions as well.
Yes.
Thank you both, and thank you to everyone for sending the questions in and for taking part and commenting during the course. It's been really interesting. Thank you.